It's my pleasure to introduce our next speaker, Nico Abuid, application scientist at ACROBiosystems. To give you a little bit of background to Nico himself, he's a West Coast field application scientist for ACROBiosystems. He's a trained biomedical engineer whose doctor focus was type 1 diabetes cell therapy. And at ACROBiosystems, Nico is working with leaders in both industry and academia to support their research needs. Nico is also part of a team that helps to guide in the use and development of recombinant proteins that can be used for COVID-19, CAR T cell therapy, as well as many other immune and autoimmune conditions. Following the talk, we are opening the floor to you, the audience, for our exclusive Q&A session where we'll be putting forward your questions to our speaker. And the best thing is you don't have to hold on to your questions, because if you think of a great question midway through the presentation, all you have to do is pop them into the box on the left-hand side of your screen and press send. And don't worry if we don't read out your question, or you think of a question whilst we're watching this on demand. All of the questions that don't get answered will be contacted offline with an answer. If you do have any technical issues during the event, you can click the question mark in the top right-hand corner of your screen to request the team's support. Before we begin, I also want to quickly remind you to navigate over to the literature section where you can download the latest resources from the sponsors of today's webinar. So without further ado, I'll now hand over to Nico.
Thank you very much for that very kind introduction. So as you mentioned, I am here to talk to you about ACROBiosystems' journey and collaborations with immune therapy companies and vaccine development leaders. My name is Nicolas Abuid, and I'll start my presentation now. So first of all, I would like to talk to you a little bit about ACRO. ACROBiosystems was founded by Mike Chen in 2010. He started this company as a CRO, and as he progressed his work, he started to express some different proteins, one of them was HER2. People saw the quality, the stability, the high purity of the proteins, and started to show a lot more interest. So that's when he started to expand his catalog to other different proteins. Now we're committed to accelerating the process of target therapeutic drug development for different applications, including in vitro and in vivo testing, antibody humanization, clinical trials, ELISA, NIH validation, etc. Now we have over 2,000 different products for immune therapy, neurobiology, and COVID-19. We are located in Newark in the US and Beijing in China, but we also have satellite offices in San Jose, California, Boston, Massachusetts, Hangzhou, and Shanghai. In addition to this, we also have a global presence beyond China and the US in Canada, Latin America, Europe, and we're expanding to Africa. So we have now more than 4,000 different customers in more than 60 countries. And our main collaborators are researchers in biotech, pharmaceutical companies, and universities. As I mentioned before, we started with some enzymes, and we moved towards immune checkpoints, targets of CAR T cell therapy, cytokines, targets for ADC and bispecific antibody. Following this, we worked on full-length transmembrane proteins, which are difficult to express proteins. And since the beginning of the pandemic, we have been working on SARS-CoV-2 related products and mutants. For that reason, I will skip most of this topic into more into SARS-CoV-2 proteins. So we all know that throughout the pandemic, we started taking some measures to prevent the spread, including the increasing the manufacturing of PPE and enforcing mask and social distancing mandates, and studying some antibody therapeutics or therapeutics such as remdesivir, dexamethasone, which is a potent anti-inflammatory reagent, and monoclonal antibodies. However, as we all know, one of the most effective pandemic solutions is a vaccine, as we have seen before with the polio, the MMR, the smallpox, and the flu shot. Since the beginning of the pandemic, we've seen, since the release of the vaccines, we've seen a decrease in cases with an increase in vaccination. And as we increase the number of vaccines, we also decrease the spread and the mutation rate. For that reason, I would like to point out a few of different vaccine development strategies, including inactivated vaccines such as the CoronaVac, the proteins of unit vaccine such as the Novavax, virus vector, as we mostly know, Janssen and AstraZeneca, mRNA and DNA vaccines as is one of the most administered vaccines, the Pfizer, Moderna, and Cycle D with the DNA vaccine. For many of these different strategies, we have collaborated, and I will walk you through some of the collaborations we've done with this vaccine development strategies. Regardless of the vaccine strategy, there is a common workflow to follow when developing the vaccine. This starts with production and QC, moving forward to preclinical studies, and finally to clinical trials 1 through 3. So I will, with that, I would like to move and talk to you about some of the different proteins, kits, and products for vaccine developers and in therapy developers as well. We have a spike protein as well as ACE2 proteins, which we provide very high purity, including SDS page and SEC MALS report. In addition to this, we also provide support for customers and collaborators doing bioactivity, ELISAs, BLI, and SPR. And in addition to this, we also provide protocols to support and facilitate the research of our collaborators, which these protocols include the concentration, the different buffers, and appropriate conditions to perform your studies. In addition to the different proteins that we develop, we also have ELISA kits used to quantify the content of antigen, such as a spike protein titer assay kit, RBD titer assay kits, and spike protein ELISA kits for vaccine development, which have a high potency against different variants of concern. We also help with efficacy evaluation using indirect ELISA kits to detect titer anti-SARS-CoV-2 mouse IgG antibodies for detection of IgG antibodies in serum, plasma, and in vitro. Finally, we also have competitive ELISA kits for detection of titer anti-SARS-CoV-2 neutralizing antibodies with the respective neutralizing antibody control that can be used to detect spike S1 protein in a sandwich ELISA or LFA assay. Now, once you're done with your production QC, the next logical step is to work on preclinical studies. For this, we offer kits to study monkey IgG antibodies and the respective antibody controls that can be used to detect spike S1 protein in sandwich ELISA or LFA assay. As I mentioned before, we also provide quality reports such as SDS page and SEC MALS with every one of our lots to ensure lot-to-lot consistency and minimal variability. Finally, once this is concluded, we also have clinical trial kits, human antibody IgG serological assay kits for spike RBD, spike trimer, and spike S1 that can be used to detect antibody in serum plasma. And we also provide toxicity evaluation reports to study the antibody subtype from IgG 1 through 4 with minimal cross-reactivity with other coronaviruses. Now that I talked to you about the different products, I want to point out some of the most common collaborations we've done with industry. The most commonly known one is the collaboration with the ACROB, where they use a recombinant S1 spike protein for detection of antibodies can you stop? I'm going to start again with this site. One of the most commonly known collaborations was the ACROB using recombinant S1 spike protein to measure antibody titers via ELISA. In this study, they use a recombinant S1 spike protein from ACROBiosystems to study the antibody response after DNA vaccination in biopsy mice, guinea pigs, and rabbits, where they either were immunized at week 0, 2, and 4 with the DNA vaccine, and sera was collected at day 28 and day 42 and evaluated for SARS-CoV-2 S1 spike IgG antibodies. Another well-known application of our products is the BNT162b1 Pfizer vaccine that they used our recombinant RBD to study the robust immunogenicity that was observed after vaccination with the Pfizer vaccine. RBD binding IgG concentrations were detected at day 21 after the first dose and was substantially increased 7 days after the second dose, which was given at day 21. And in addition to all the work we did with initial vaccine developers, we also worked on different COVID variants. For that reason, I prepared a small graph showing the trajectory of the vaccine or of the variants throughout the pandemic since we started back in December 12, when the WHO declares COVID-19 pandemic in March, and all the different variants of concern. For all these variants of concern, we have developed products, including for the Omicron, which we were able to get these products out in almost record time, and help with diagnostic and vaccine developer to understand this the variant. In addition to this, we worked with the Alpha variant, the Beta variant, Gamma, Lambda, and Delta variants by developing products such as spike proteins, RBD, S1, with different formats, HisTag versus FCTag versus ABTag, and different components of the COVID-19 variant. In addition to some of the less prominent variants of concern, which you can find all of this in our website. Another important table that you can find in our newsletters and website is this, showing the different variants and the transmissibility, immune evasiveness, and vaccine effectiveness. As shown here, there is the vaccine effectiveness against Omicron is not very well characterized. And for this reason, we collaborated with a paper in Nature by that studied the effectiveness of the coronavac vaccine against the Omicron variant and different variants of concern. The coronavac, a beta-propylactin inactivated SARS-CoV-2 vaccine, was approved for emergency use and recommended for booster vaccine in older people by the WHO. The serum specimens were collected from two groups, one that had two vaccines and one that had two vaccines and a booster. And the results showed that the vaccines, the subjects with 3 doses had a higher protection against wild type Delta and Omicron compared to subjects with 2 vaccines. and none of these volunteers recruited for vaccination were infected with SARS-CoV-2 before the study. In following this, the researchers in this study used our inhibitor screening kit to study what are the different neutralizing antibodies that are developed, that are released by memory B cells, and try to find if the immune evasive mutation and how to neutralize this mutation. After separating a few of this, they use our BLI, our products and our BLI protocols to study some of the most prominent neutralizing antibodies, and if a combination of this would help neutralize Omicron. And if you want more information of this, you can ask me questions, and I'll be more than happy to put you in contact with some of the authors of this paper, as Mike Shen, our CEO, is actually also one of the co-authors of this study. And with this, I would just like to open the floor for some questions. But before this, we are hiring at all levels, so we have an amazing team, and you can feel free to join us. And if you do have more questions about working at ACRO, you can contact us.hr@acrobiosystems.com for more information. And stop.
Perfect. Thanks very much, Nico. That was a great presentation, and I'm sure that our audience now has a whole host of questions for you. So we'll now start your Q&A session. Remember, if you'd like to ask Nico a question, you can type this to us in the question box on the right-hand side of your screen and click send. We'll answer as many questions as possible during the time that we have available. We've already had several questions submitted, so I'll move straight on to these. The first question I can see is, what are the new variants of concern that your team is working on?
I'm glad you asked. We actually have some products out for the Deltacron, the BA.2, 0.3, and 0.4. So we've been asked a lot about these different variants of concern, and we have some products out for to complement their studies.
And the second question I can see is, are there any ACE2 therapies your collaborators are working on?
We actually do have some customers and collaborators that are working on ACE2 therapies, and we also do support work for this. We have some products to support this research.
Nice. And how long does it take your team to develop the new Omicron products since they came out?
Actually, yes, since it came out back in November 26th of 2021, we started to work on getting this highly mutated RBD and Omicron and all the different components. And I believe it was around 3 to 4 weeks to get all these products out.
Wow.
And start providing proteins, H2 spike proteins, and proteins to supplement the studies of our collaborators and customers.
Fantastic. And the final question I have are, what are the new product lines your company is working on?
I'm glad you asked. Since we started working in cancer therapy, moving to COVID-19, now we actually have a new line of neurobiology targets. We want to expand to different fields of research, and since we already have experience with glioblastoma and related targets, we wanted to support the neurobiology research as well.
Fantastic. Well, unfortunately, they are all the questions that we have time for today, but thank you, Nico, firstly for your work, secondly for your incredible presentation, and thirdly for just taking the time to be with us today.
Thank you very much.
And just to remind everyone, any questions that we didn't have time to get to will be answered offline as soon as possible. And you can still continue to ask questions even when you're watching this webinar on demand.